Hi, welcome back to LPHumananatomika.
Mitochondria, a very important cell organelle in the cytoplasm, are mobile organelles. They are inherited from mother. Please note, we do not get mitochondria from father. For that matter, not only mitochondria, all other cell organelles except nucleus are inherited from mother. Each active cell presents numerous mitochondria which are vesicular membrane-bound bodies. They are in the form of either filamentous or rod or spherical or thread-shaped structures. These bodies can be seen under light microscope after staining with special stains. Electron microscope reveals that each mitochondrion consists of two membranous walls, the outer and the inner membrane, and two chambers, outer and inner chamber. Each membranous wall presents unit membrane, but mitochondrial membranes are very rich in proteins and are separated by an intermembranous space that is nothing but outer chamber. The outer membrane is a smooth, continuous membrane which covers and protects the mitochondrion. It separates the mitochondrion from the cytosol. It is permeable. The outer membrane allows small molecules to be exchanged between the cytoplasm and the intermembrane space. This exchange occurs through proteins called porins, which are present in the outer membrane. The outer membrane also hosts a number of enzymes with a wide variety of functions. Inner membrane is folded or projected inwards to form incomplete partitions known as cristae or cristae mitochondrialis. The membrane shows two faces. The outer face directed towards outer membrane is called C phase or cytosol phase, and the inner phase is called M phase or matrix phase. On this M phase, a number of ribosomes and special enzymes for phosphorylation of ADP to ATP in the form of knob-like structures are impregnated. The inner mitochondrial membrane is completely impermeable even to small molecules, with the exception of molecules like oxygen, carbon dioxide, water, ATP synthase and enzymes. It is postulated that with the progress of evolution, the inner walls of the mitochondria are derived from attenuated bacteria, which are drawn into the cytoplasm of the animal cells and undergo symbiotic life to complete aerobic respiration of the invaded animal cell. Moreover, mitochondria divide by fission similar of that of bacteria. This endosymbiont theory is supported by many findings. For example, mitochondria have their own DNA and ribosomes. The mitochondrial genome became smaller and smaller during the course of evolution. In humans, it is still containing 16,569 base pairs which code for 2 RNAs, 22 tRNAs and 13 proteins. Only these 13 proteins are produced in the mitochondrion. Outer chamber is the space between the outer and inner membranes. It is 60 to 80 angstroms wide, which is filled with a fluid. The inner chamber present in the interior of each mitochondrion is filled with fluid which is gel-like substance called as the mitochondrial matrix, which contains circular form of DNA, RNA and important enzymes like that of bacteria. Chemically, mitochondria is composed of 73% of proteins, 25 to 30% of lipids, 5% of RNA and small amount of DNA. The enzyme complexes are more. The number and shape of the mitochondria as well as the numbers of cristae they have can differ widely from cell type to cell type. For instance, mature red blood cells do not have mitochondria, whereas liver cells can have more than 2000 mitochondria. Cells with high demand for energy tend to have greater numbers of mitochondria. Around 40% of the cytoplasm in heart muscle is taken up by mitochondria. Even with one type of tissue, the shape of the mitochondria can vary depending on their functional status. In sperm cells, the mitochondria are spiraled in the midpiece and provide energy for tail motion. Cristae are more in tissues with intensive oxidative metabolism like heart muscle. The most important role or function of the mitochondria is to produce the energy currency of the cell, the ATP, through cellular respiration and to regulate cellular metabolism. Mitochondria convert chemical energy from the food we eat, breaks the food and turn it into energy form called kinetic energy by forming ATP that can be used by the cell. Because of this, it is called as powerhouse of cell. This process is called oxidative phosphorylation. Mitochondrial antiviral signaling protein plays a key role in the innate response to viral infections, helping to induce antiviral and anti-inflammatory pathways. The mitochondria in liver cells have enzymes that detoxify ammonia. Mitochondria help the cells to maintain proper concentration of calcium ions within the compartments of the cell. Mitochondrial calcium exchange is the flow of calcium in and out of a cell's mitochondria, a process which is important in metabolic regulation and cell death. Calcium is vital for a number of cellular processes. For instance, releasing calcium back into a cell can initiate the release of a neurotransmitter from a nerve cell or hormones from endocrine cells. Calcium is also necessary for muscle function, fertilization and blood clotting. Because calcium is so critical, the cell regulates it tightly. So how does the mitochondria act in this? Mitochondria play a part in this by quickly absorbing calcium ions and holding them until they are needed. Cell death, also called apoptosis, is an essential part of life. As cells become old or broken, they are cleared away and destroyed. Mitochondria help decide which cells to be destroyed. Apoptosis is the highly controlled process which is used by multicellular organisms in a number of biological processes, including intrauterine development, cleaning damaged cells, and maintaining cell numbers. Disturbances to this regulation are associated with the development of diseases such as cancer and tissue damage following stroke. Mitochondria are thought to play crucial roles in the maintenance of pluripotency, differentiation, and reprogramming of induced pluripotent stem cells. The generation of reactive oxygen species by mitochondria has been shown to regulate somatic stem cell fate. An increase in ROS is associated with a decrease in the regeneration potential of human mesenchymal stem cells and a move towards progenitor commitment and differentiation. It also helps in cell signaling. Cellular respiration, a multi-step process, is essential for proper functioning of the body, and dysfunction can contribute to a variety of diseases ranging from diabetes to Parkinson's disease to rare genetic disorders. So when mitochondria stop functioning, the cell they are in is starved of energy. Depending on the type of cell, symptoms can vary widely. As a general rule, cells that need the large amounts of energy, such as heart muscle cells and nerve, are affected the most by faulty mitochondria. Abnormal mitochondrial DNA may produce muscular weakness and degenerative disease of central nervous system due to failure of oxidative metabolism. This is known as mitochondrial cytopathy syndrome. Disturbances in the process of apoptosis regulation leads to development of diseases such as cancer and tissue damage following stroke. This handful of examples gives just a snapshot of the mighty influence that mitochondria can have on cell function and health. The continued discovery of these intricacies and the mechanisms followed in mitochondrial dysfunction should lead to the development of new and improved therapies for a wide range of diseases. Lysosomes, component of cytoplasm of cell, are lipid bilayer thick-walled membrane-bound spherical vesicles. A lysosome has a specific composition having membrane proteins and luminal proteins. Lysosomes contain hydrolytic enzymes, namely proteases, lipases, and acid phosphatases. So we know lysosomes contain a variety of hydrolytic enzymes. These enzymes, when set free from the lysosomes, can break down many kinds of biomolecules it engulfs, including peptides, nucleic acids, carbohydrates, and lipids, and may digest certain substances which originate within the cytoplasm or substances introduced into the cell from outside. Before a cell dies due to lack of oxygen or for other reasons, the lysosomes act as autophagic vesicles and destroy all organelles of the cytoplasm. Hence, they are known as the suicidal bags of the cell. The lysosomes, rich in glycoproteins, are derived from the mature phase of Golgi apparatus as primary lysosomes. In the process of wear and tear of the cell, functionless pieces of mitochondria, Golgi apparatus, and endoplasmic reticulum coalesce with the lysosomes and undergo digestion. Such fused bodies form secondary lysosomes and are known as cytolysosomes. Thus, the lysosomes are endowed with the function of removing the damaged structures or degradation remains of the cytoplasmic organelles. This process is called autophagy. After death of animal, unless the animal is fixed by fixatives, the lysosomes rupture and autolyse, which is nothing but enzymatic digestion of cells by the action of its own enzymes of the cells takes place. So this process is called as autolysis. In healthy cells, the lysosomes are protective in function by destroying certain bacterial invaders acting as heterophagic vesicles. The lysosomal enzymes degrade a variety of detrimental substances within the cell. The congenital absence of particular enzymes results in accumulation of their substrates within the cells, producing storage diseases like Tay-Sachs disease and the Gaucher's disease. Such lysosomes are abundant in macrophage cells and granular leukocytes. Some of the insoluble remains of lysosomes after autophagy and heterophagy form residual bodies which are retained permanently as senility pigments made of lipid-rich lipofuscin, hence called lipofuscin pigment. These pigments are more found in old age within the nervous system. Besides degradation of polymers, the lysosome is involved in various cell processes including secretion, plasma membrane repair, cell signaling, and energy metabolism. Centrosome, another component of the cytoplasm of the cell, in Latin means central body. It serves as the main microtubule organizing center of the cell as well as a regulator of cell cycle progression. Each cell which is capable of division during interphase, the period in which a cell is not actively dividing, possesses one centrosome within the cytoplasm, close to the nuclear membrane. Centrosome includes a pair of centrioles placed perpendicular to each other in a dense cytoplasm. So, in other words, the dense region of cytoplasm containing pair of centrioles is known as the centrosome. The two centrioles are surrounded by an amorphous mass of protein termed the pericentriolar material. Wall of each of these centrioles consists of 9 longitudinal bundles of microtubules in a cylindrical arrangement embedded in fibrillar materials. Each bundle has a 3 microtubule arranged parallel fibers arranged each other. A single centriole includes a total of 27 microtubules running from end to end. The arrangement of these microtubules when looked from one end gives cartwheel appearance. In centrosome replication during S phase of the cell cycle, during the prophase in the process of cell division, the centrosomes migrate to opposite poles of the cell. The centrioles help synthesize the continuous microtubules called the achromatic spindle by the linkage of soluble cytoplasmic protein known as tubulin. The two centrosomes are separated from each other by the growing microtubules by the achromatic spindle and occupy the opposite pole of the cell. By the end of the cell division, each of the two new daughter cells derived from the cell division contains one centrosome with two centrioles set at right angles to each other. The centrosome is copied only once per cell cycle so that each daughter cell inherits one centrosome with two centrioles. The centrosomes interact with the chromosomes to build the mitotic spindle. The mother centriole, the older of the two in the centriole pair, has a central role in making cilia and flagella of newly formed daughter cell, and it is a regulator of cell cycle progression. Replication of a centrosome is similar to DNA replication in two aspects: the semi-conservative nature of the process and the action of CDK2 as a regulator of the process. The mother centriole helps in the accumulation of materials required for the assembly of the daughter centriole. Any malfunction in the pathway of replicating the centrosomes leads to aberrant number of chromosomes, which is shown to have to be associated with cancer. Peroxisomes, other component of the cytoplasm in the cell, are single membrane bound vesicles about 0.5 to 1.5 micron meter in diameter, hence called microbodies. These are made of a phospholipid bilayer with many membrane bound proteins. They contain oxidative enzymes and sometimes contain crystalline core. Peroxisomes are present in most of the nucleated cells and are more numerous in hepatocytes and renal tubules. Depending upon the energy requirements of the cell, peroxisome vary in shape, size and number. It plays important role in energy metabolism, lipid biosynthesis and oxidative enzymes help detoxification of various substances like reactive oxygen species. They participate in oxidative pathways like beta oxidation of fatty acid chain. They generate hydrogen peroxide, and excess amount of hydrogen peroxide is broken down by the enzyme catalase, and it also helps in signaling of cells. Microtubules are filamentous structures made of soluble tubulin proteins which are contractile in nature. All cells possess microtubules which may be organized or dispersed. Cilia, flagella and centrioles are composed of microtubules which are organized. The dispersed microtubules act as skeleton of the cell and help in transport of various substances including the macromolecules throughout the cytoplasm. Since the microtubules are composed of contractile proteins, they are concerned with movements of cilia, flagella and achromatic spindles through which centrioles are pushed to opposite poles of the cell. At least 3 sites are available in the cytoplasm which act as microtubule organizing centers. The first one is the centriole for the continuous microtubules of the spindle, kinetochore of the chromosome for chromosomal microtubules, basal bodies of the cilia for the growth of ciliary microtubules. Colchicine, a chemical substance, arrests the cell division at metaphase by combining with tubulin protein and preventing the formation of the achromatic spindle. The colchicine is used in genetic labs. Cilia, flagella and centrioles are organized type of microtubules. The dispersed microtubules are present as network of tubules beneath the cell membrane and act as skeleton of the cell and help in transport of the substances including the macromolecules throughout the cytoplasm. Filaments and microfibrils are ultra microscopic network of filamentous structures which are different from microtubules. Some of the filaments are more dense beneath the cell membrane, forming the cell web and act as internal support to the cell. Some filaments are present in central core of the microvilli, while others form actin filaments of the contractile muscles.
If you like the video, hit the like button. If you have not yet subscribed, subscribe to the channel and hit notification button. Join my private Facebook community where like-minded students will support, discuss, encourage and push each other towards reaching the goal. For link, visit the description area. Thank you for watching.